To continue with the series of photosynthesis, today in the fourth lecture, we will talk about, or we will try to learn about the C3 and C4 plants. Now, what are C3 and C4 plants? C3, that is 3 carbon and 4 carbon plants. Now, C3 plants are mostly the plants, most of the plants that you see around you, they will be called as C3 plants. Why they are called as C3 plants? Because the first stable product in Calvin's cycle during photosynthesis in such plants is a 3-carbon compound, phosphoglyceric acid. If you can recall Calvin's cycle, there you will see that the first stable product is always a 3-carbon compound, phosphoglyceric acid. Hence, these plants are called as C3 plants. Most of the plants that you see around you will be C3. Now, C4 plants, on the other hand, are some tropical grasses like corn, sugarcane. That is, mostly monocotyledonous plants fall under the category of C4 plants. Why they are called as C4 plants? Because in them the first stable product is a 4-carbon compound, oxaloacetic acid. It is a 4-carbon compound. We will talk about it now in details. So this is the major difference between C3 and C4. Most plants around you, now some tropical grasses like corn, sugarcane, maize, this comes under C4 plants. Now here you can see the pictures. Okay, so these are wheat, soybeans, potato plant, rice. This comes under C3 plants. This comes under C3 plants. Then you have, these are the C4 plants. Okay, these will come under C4 plants. Okay, now the fixed carbon in Calvin's cycle attach carbon dioxide to RUBP. Fixed carbon in cytoplasm attach carbon dioxide to PEP. PEP means phosphoenol pyruvate. So carbon dioxide, once it's inside the cells, it attaches to ribulose biphosphate in C3 plants, whereas in C4 plants it is accepted by phosphoenol pyruvate. The main enzyme is Rubisco. Here the main enzyme is phosphoenol pyruvase. This is the main enzyme. Most energy efficient method. Halfway between these two, then it loses water through photorespiration, loses less water. Now photorespiration, we will talk about it in the next upcoming lecture. Now it loses less water. The loss of water is less. Now here you can see these are the internal structures of C3 and C4 plants. This is C3 and this is C4 plants. Due to the internal internal structure differences, what happens is the process also differs. Internally, the mesophyll cells, the arrangement is different. The arrangement is different in C3 and C4 plants, and as such, what happens is the process of photosynthesis also differs. So here you can see the C4 plants mainly. Okay, the C4 plants mainly, the C4 plants, what happens is you will see the C4 plants, so the C4 plants, they have the mesophyll cells. These are the mesophyll cells, is the TS of the leaf, LS of the leaf. So you will see these are the epidermal cells, and these also are the lower epidermis. Then these are the mesophyll cells. Then around the vein. Now vein means the vascular tissues. Now around the veins you will find spatially arranged bundle sheet cells. These are the bundle sheet cells that are arranged. Okay, these are the bundle sheet cells that are arranged around the vascular tissues. And hence what happens, this type of arrangement is also known as Kranz anatomy. Okay, this is also called as Kranz anatomy. These are the bundle sheet cells, very specifically meant only or present only in C4 plants. In some C3 plants also you do find bundle sheet cells, but these are very limited or very lesser in number or almost negligible. But in C4 plants you will find very prominent bundle sheet cells. Now bundle sheet cells, attached to them are the mesophyll cells which you see here. These are the mesophyll cells attached to them immediately. So in a circular manner you will find. Now what happens in case of C3 plants, the entire process of photosynthesis occurs within the chloroplast of the mesophyll cells. But in case of C4 plants, the process of photosynthesis gets distributed between the mesophyll cell and the bundle sheet cells. The main reaction, the major part of the reaction happens in bundle sheet cells and in the mesophyll cells, and in the bundle sheet cells the production of glucose, the main production of glucose takes place in the bundle sheet cells. We will see that in details now. So you can see here in C3 plants, okay, this is the C3 plants. What happens? Carbon dioxide is taken in. This is taken in. This is a mesophyll cell. Okay, this is a mesophyll cell. So through the stomata, carbon dioxide enters, accepted. Now of course light reaction has already taken place, and then in the stroma what will happen? The Rubisco, with the help of Rubisco, RUBP will take up carbon dioxide, carry out the Calvin cycle and produce glucose. During light reaction, the oxygen release will be removed out. So that is the process of photosynthesis in C3 plants. Now in C4 plants, like say corn, what will happen? The carbon dioxide is taken in. First it is accepted by the mesophyll cells. In the mesophyll cells, it is accepted by PEP, phosphoenol pyruvate, okay, and then after that it is converted to compound, okay, malic acid and so on. It's converted. That compound then moves into the bundle sheet cells, okay, where the concentration of carbon dioxide increases. The concentration of carbon dioxide increases here, and Rubisco is not present in mesophyll cells. Rubisco is present only in the bundle sheet cells. Unlike C3 plants where Rubisco is present in the mesophyll cells, in the C4 plants Rubisco will be present only in the bundle sheet cells. As such, Calvin cycle is possible only in the bundle sheet cell. In mesophyll cell, the Calvin cycle cannot take place. So production of glucose will take place only in the bundle sheet cells, after which it will move to the phloem. Now the question might arise in your mind, why this difference? Okay, why this difference? This difference is mainly to increase the concentration of carbon dioxide so that there is more production of glucose. There is more production of sucrose or carbohydrates. That is the main reason why C4 plants are they have been evolved. So here you can see the chemical reactions. In C4, carbon dioxide from the atmosphere, it enters into this yellow is mesophyll cell. This is the mesophyll cell. Carbon dioxide enters, accepted by phosphoenol pyruvate PEP, which is already present in the mesophyll cell of only C4. Only corn-like plants you will not find in other plants of C3 category. So here PEP accepts carbon dioxide, then it reacts to form a compound oxaloacetic acid or oxaloacetate. It is formed. after which it gets converted to malate or malic acid. This malic acid will now be transported into the bundle sheet cells. This is the bundle sheet cell. So it will be transferred to the bundle sheet cells. Now this malic acid, it will now split. It will split into carbon dioxide again and pyruvic acid. So you can see the malic acid that got transported splitted into carbon dioxide and pyruvic acid. Now this carbon dioxide concentration has increased. It will be accepted by ribulose biphosphate RUBP, which is already present in the bundle sheet cells and also exclusively present only in the bundle sheet cell, not in the mesophyll cells of C4. RUBP will accept carbon dioxide, and the normal Calvin cycle, the three-step process, that is carboxylation, reduction and regeneration will take place, resulting into formation of carbohydrates. Okay, that is the glucose. Now the pyruvate that was formed here, this pyruvate will now again be transported back to the mesophyll cells for production of phosphoenol pyruvate, so that there is continuous production of phosphoenol pyruvic acid. So this way the C4 cycle continues. I repeat, carbon dioxide is absorbed by PEP. It's converted to oxaloacetic acid and then to malic acid, which gets transported into the bundle sheet cells to split into carbon dioxide and pyruvic acid. Carbon dioxide will be accepted by RUBP. Once this RUBP accepts carbon dioxide, it will start with the normal Calvin cycle to produce carbohydrates or glucose, which will now be used for giving energy to the plant. In the meantime, the pyruvic acid formed in the bundle sheet cells will be transported back to the mesophyll cells, where it will form PEP. Why PEP is required? So that it can continue to take up carbon dioxide. And one more point can be added here. Now the light, this is only for the dark reaction or biochemical phase. Now the photochemical phase, what happens is, in the photochemical phase will take place only in mesophyll cells. Now vascular bundle sheet cells cannot carry out light reaction. Why? Because here there is total absence of grana. Grana is not present in the chloroplast of the bundle sheet cells. It is present only in the mesophyll cells. So light reaction cannot take place in the bundle sheet cells. Once more, the differences between C3 and C4 plants. Now I think it will be better for you to understand. So photosynthesis occurs in mesophyll cells only. Photosynthesis occurs both mesophyll and bundle sheet cells. So here the point to remember is the photochemical phase in mesophyll cells only. Biochemical phase, a part in mesophyll cells and completed in bundle sheet cells. Carbon dioxide acceptor is RUBP. Carbon dioxide acceptor is phosphoenol pyruvate. They do not have Kranz anatomy. That means they do not have the regular circular arrangement of bundle sheet cells, whereas in C4 they have Kranz anatomy. Photorespiration occurs. Photorespiration does not occur. Now photorespiration is a process where the Rubisco enzyme, the Rubisco enzyme, which is present along with RUBP, sometimes it does not act only to take up carbon dioxide, but it also acts to take up oxygen. If the amount of oxygen increases, then what will happen? If the amount of oxygen increases, then it will act to take up oxygen. And thereby what will happen? It will not be able to take up carbon dioxide. And so the photosynthetic process gets hampered. So that is photorespiration. So in C3 plants, photorespiration occurs, thereby hampers the photosynthetic pathway. But in C4, photorespiration does not take place. It is prevented. So that way also it is advantageous. Okay, now we will talk a little bit about photorespiration too. Now photorespiration. Okay, now photo means light, respiration, that is utilization of or absorption of oxygen and thereby release of energy. So light induced respiration, dual role of Rubisco. Now, what is this dual role of Rubisco? Now, the Rubisco enzyme, as you have already seen, Rubisco enzyme is very much important for absorption of carbon dioxide and then getting converted to phosphoglyceric acid during Calvin cycle. Now, Rubisco, the full form of Rubisco is ribulose biphosphate carboxylase oxygenase. Carboxylase oxygenase. Now, this particular enzyme, depending upon what type of gas is present, if it is getting sufficient carbon dioxide, it will help in carrying out the normal RUBP will accept carbon dioxide and then normal C3 cycle will continue releasing sugars. But if suppose in carbon dioxide concentration is less, oxygen becomes very high. Suppose oxygen becomes very high, then what will happen is Rubisco will start taking up oxygen. Okay, it will start taking up oxygen instead of carbon dioxide, and then instead of yielding sugar, it will take a different pathway which we call as photorespiration. In this pathway, there will be no formation of sugar. So I hope you are getting that. So if the plant is getting higher oxygen in place of carbon dioxide, then it will not produce sugar, which it is supposed to. Thereby the photosynthetic pathway gets hampered. So when is the possibility that plant might get more oxygen? That is during light reaction, as you have seen oxygen evolution take place. So if the oxygen evolution is higher, then Rubisco will act to carry out photorespiration and thereby hamper the photosynthetic pathway. Now, what are the advantages and disadvantages? Advantages is utilizes a part of light energy and saves the plant from photooxidative damage. So sometimes what happens, excess light energy might damage the plant cells. Oxidation of the plant cells might take place. So thereby yellowing, the chlorophyll might get damaged. So what happens? Due to this photorespiration happening, a part of light energy is absorbed during this process. So thereby the plant cells are protected. But disadvantages are it neither synthesizes sugar nor ATP synthesis takes place. So it is like it gets disadvantages that way. Release of carbon dioxide takes place with utilization of ATP. Again, carbon dioxide release takes place where carbon dioxide should have been absorbed, carbon dioxide release is taking place. So these are the disadvantages of photorespiration. That is why in due course of time, the plants have evolved to C4 plants whereby they have separated the light reaction and dark reaction into different cells, just now what we saw. So if in mesophyll cells only light reaction takes place, so it never has the chance of coming in contact with Rubisco, which is present only in bundle sheath cells. So thereby photorespiration is totally prevented. Okay, the mechanism of photorespiration. When light intensity is high, the light reactions of photosynthesis liberate more oxygen. So when light intensity is high, Rubisco favors oxygenation of RUBP, which I just now explained, resulting in formation of glycolic acid, which is a 2-carbon compound. This initiates photorespiration, inhibits photosynthesis. So it is a scavenging process helping to getting rid of assimilatory power, that is the light energy. The process happens only in C3 plants, involves chloroplast, mitochondria, peroxisomes. These are 3 cell organelles involved in photorespiration. Then what are the factors that affect photosynthesis? The 4 factors affect photosynthesis, as you can see here: light, water, carbon dioxide and temperature. So light provides energy, water, the photolysis, carbon dioxide, the main source for formation of glucose, temperature, so that there is efficient efficiently the enzymes can act. Now, limiting factors. What are limiting factors? Law of limiting factors. Rate of a physiological process will be limited by the factor which is in shortest supply. Any change in the level of a limiting factor will affect the rate of reaction. That means if a number of factors are affecting or is influencing a process, the factor which is least will be having the maximum effect. Say here lower carbon dioxide concentration, lower temperature. Higher carbon dioxide concentration, lower temperature. Higher carbon dioxide concentration, higher temperature. So the least factor, okay, the factor which is lowest, like suppose light intensity, water and carbon dioxide temperature, all the factors are equal. So equally they are getting. Now what happens? Suppose the light intensity reduces very much. Okay, the light intensity reduces very much. At that point of time, what will happen? The other factors, even if they are high, but the rate of photosynthesis will be determined only by light because it is low. Suppose light increases, intensity of light increases, and the carbon dioxide concentration as well as temperature also decreases. Okay, it decreases. Then what happens? The rate of photosynthesis now will be determined by this low carbon dioxide or this low temperature, whichever is lowest. That will determine the rate. So I repeat, law of limiting factors means when a number of factors is influencing a process, the factor which is least quantity will have the maximum effect on the rate of the reaction. That is law of limiting factors, and these are the four factors that affect photosynthesis. So that is, with that we come to the end of the series of lectures on photosynthesis.